Hello students, I am Dr. Vinod Shinde. In the previous lectures, we have discussed about the chemical disinfectants, that is phenol, alcohols, halogen, heavy metals, quaternary ammonium compounds, gaseous aldehydes, gaseous sterilizing agents and aldehydes, and soaps. So, in this lecture, we will discuss about the evaluation of disinfectant, or in other terms, it is phenol coefficient. What is the phenol coefficient, or what is the importance of phenol coefficient is the most important thing. Phenol coefficient is a technique in which we describe or evaluate a chemical as a disinfectant in reference with phenol as compared to phenol. Laboratory technique for evaluation of antimicrobial chemical agents are conducted by one of three general methods. Here we are conducting phenol coefficient method. The chemical agent is tested against a specified microorganism referred to as a test organism. To standardize antimicrobial agents, several kinds of properties are evaluated. These properties are the ability to prevent growth of microorganism, the ability to kill microorganism already grown, the spectrum of activity or a type of organism affected by that agent, that is if the chemical agent is a broad spectrum or a not. Then the toxicity or ability to be used in or on living medium. These four characteristics are evaluated for the evaluation of a disinfectant as a chemical disinfectant. Evaluation of a chemical as a chemical disinfectant. So, in phenol coefficient method, a number of different methods have been developed to measure the effectiveness of disinfectant. However, the most widely used method to evaluate disinfectant is to rate them according to their phenol coefficient. The phenol coefficient is defined as the killing power of a disinfectant against the test organism as compared to that of phenol under identical conditions. The conditions must be specified as the organism in use of the test must be similar, age of the culture must be similar, the temperature of the test must be similar, time of action of disinfectant and phenol must be similar, presence and amount of organic matter in the culture must be similar, proportion of disinfectant to the culture must be similar, and conditions or composition and reaction of transfer medium, temperature and time of incubation of the transfer medium. These conditions must be kept identical to test a chemical as a disinfectant in the chemical disinfection or phenol coefficient method. A specific official test method based on principal outline in the first procedure is Aoac phenol coefficient method, sometimes called as FDA method. Aoac is nothing but Association of Official Agricultural Chemists, and FDA is Food and Drug Administration. This procedure is suitable for testing disinfectants miscible with water and exerting their antimicrobial action in the manner similar to that of phenol. In the procedure for phenol coefficient, this coefficient method is used for identifying a chemical as a disinfectant in reference with phenol, in comparison with phenol. So, the procedure is to a series of dilutions of disinfectant being tested, that is 5 ml per tube. Dilution of phenol was dilutions of disinfectant to be tested was first prepared, and in each tube 5 ml of phenol 5 ml of disinfectant was added. To these tubes 0.5 ml of 24-hour-old culture of the test organism is added. At the same time, similar addition in the same amount are made to the series of dilutions of phenol also. All the tubes, disinfectant plus organism and phenol plus organism, are placed at 20 degrees Celsius water bath, and at intervals of 5, 10 and 15 minutes, subcultures are made with a loop transfer needle into a sterile tube of medium. That is the organism plus disinfectant and organism plus phenol. These tubes are first dilutions of phenol and dilutions of disinfectants are made. In that 0.5 ml of 24-hour-old culture of a test organism is added, and then the tubes were incubated. The disinfectant plus organism tube and phenol plus organism tube were incubated at 20 degrees Celsius water bath. And at interval of 5, 10 and 15 minutes, subcultures or some subculture was taken from the loop transfer needle into a sterile tube of medium. The inoculated subculture tubes are incubated and subsequently examined for growth after 24 to 48 hours. The greatest dilution of the disinfectant killing the test organism in 10 minutes, but not in 5 minutes, is divided by the greatest dilution of phenol showing the same result, that is killing of the test organism in 10 minutes. The number obtained by this division is the phenol coefficient of the substance tested. It should be taken care that no single microbiological test method is suitable for evaluation of the germicidal chemical or chemical as a disinfectant for all applications recommended. Therefore, one must exercise care in selecting the test method for a specific chemical agent. Now, below is the example for calculation of phenol coefficient. If you have taken dilutions of disinfectant, that is 1:100, 1:125, 1:150, 1:175, 1:200, 1:200, and phenol coefficient dilutions were 1:90 and 1:100. Now, in the case of this example, the dilution showing growth, no growth, was last minimum dilution, that is last dilution, that is showing no growth in 10 minutes as suggested. That is the greatest dilution showing dilution showing disinfectant killing of the test organism is 1:150 in 10 minutes, and similar in phenol coefficient is 1:90. So, we have taken phenol coefficient as division of 150 by 90, that is 1.6. The disinfectant in the example is having a phenol coefficient of 1.6. This is the phenol coefficient method which is used for the standardization of new chemicals or unknown chemicals as a disinfectant or used as a disinfectant purpose. So, this is the method of phenol coefficient. Thank you. We will discuss sterilization by physical agents in the next lectures. Thank you and all.